Anaerobic respiration, yogurt making. Today we will discuss the process of yogurt making. आज हम लोग yogurt making के process के बारे में discussion करेंगे और इस discussion के बाद आप लोग anaerobic respiration क्या है उस process को समझ पाएंगे और समझा भी पाएंगे. So today we will discuss the process of yogurt making, and at the end of the discussion you all will be able to explain the process of anaerobic respiration and what kind of products are formed. And at the same time, you will also be able to identify anaerobic respiration taking place in the production of biogas, bread making, as well as in the wastewater treatment plant. So, let us make yogurt today. आज हम yogurt बनाना सीखते हैं. So what is yogurt? Yogurt is cultured milk, and it is made with the help of bacteria Lactobacillus bulgaricus and Streptococcus thermophilus. So how can we make yogurt? We can make yogurt in just simple 7 easy steps. हम yogurt 7 easy steps से बना सकते हैं. सबसे पहले हमें क्या करना है? We have to take milk. This can be low fat milk, skimmed milk or whole milk. फिर second step में हमें क्या करना है? We have to heat the milk to around 180 Fahrenheit. हमें कैसे पता चलेगा कि जो मिल्क है वो 180 Fahrenheit तक गर्म हो चुका है? In order to test the temperature of the milk, we should use a digital cooking thermometer. As you can see here, this is a digital cooking thermometer which is used to test the temperature of the cooked food or while cooking the food. पहले मिल्क को हम गर्म करेंगे 180 फ़ारेनहाइट तक, फिर उसको हम ठंडा करेंगे 115 फ़ारेनहाइट तक. After heating the milk up to 180 Fahrenheit, we should cool it to 115 Fahrenheit. जब टेम्परेचर मिल्क का around 115 Fahrenheit होगा, then we have to add the starter. अब ये starter क्या है? यहाँ देखते हैं. Starter ऐसे पैकेट्स में मार्केट में बेचे जाते हैं. We can purchase these packets from the market and you have to see the label. हमें label देखना होगा कि जहाँ लिखा होगा yogurt cultures, L. bulgaricus and S. thermophilus. L. bulgaricus is Lactobacillus bulgaricus and S. thermophilus is Streptococcus thermophilus. So yogurt starters contain yogurt cultures. They contain the bacteria Lactobacillus bulgaricus and Streptococcus thermophilus. So this bacteria will help to convert milk into yogurt. और अगर हम ये starter नहीं purchase करते हैं, तो we can use the old yogurt that we have at our home as a starter. Then we have to leave the milk to incubate for 8 to 10 hours. फिर हमें 8 से 10 घंटे तक इस मिल्क को ऐसे ही छोड़ देना है. Then we have to, after 8 to 10 hours, we should cool the yogurt and remove the whey, which is the water that we find when yogurt has been formed. So we can remove the whey, or we can just mix the whey with the clumped yogurt, thick yogurt. And in order to increase the longevity, the shelf life of the yogurt, we can put it in the refrigerator for up to 2 weeks. So these are simple 7 steps for making yogurt. But what do we learn from this process? We learn that we human beings use microorganisms to prepare food items for us. And here we are using bacteria. So what happens when we use bacteria, Lactobacillus bulgaricus and Streptococcus thermophilus, to convert milk into yogurt? How do this bacteria convert milk to yogurt? The process of conversion of milk to yogurt is called lactic acid fermentation, and it occurs in the absence of oxygen. But how? In the absence of oxygen, the milk sugar lactose breaks down to pyruvate. Then pyruvate forms lactic acid. This lactic acid creates an acidic condition in the milk, and this acidic condition leads to the clumping together of a protein called casein, which is present in the milk. When this protein starts to clump together, then we say yogurt formation has started, and when they start thickening, then yogurt is formed. This is the simple process of explaining the formation of yogurt from milk. So what happens here? The lactose sugar of milk breaks down to pyruvate. Pyruvate forms lactic acid. This lactic acid creates an acidic condition, and because of this acidic condition, the casein protein of milk start to clump together and yogurt formation occurs. Now, you may be wondering, does milk not contain oxygen? Yes, milk does contain oxygen. Then how does the fermentation occur in the absence of oxygen? Where does this oxygen go? So milk does contain oxygen. However, the bacteria Streptococcus thermophilus, when added to milk, it uses all the oxygen and creates an anaerobic environment. So when we say anaerobic environment, it means there is absence of oxygen. And this environment is created by Streptococcus thermophilus added to the warm milk. When Streptococcus thermophilus has created an anaerobic environment, now Lactobacillus bulgaricus bacteria starts converting lactose sugar to lactic acid and proteins present in the milk to amino acids. The presence of lactic acid and amino acid creates an acidic environment, and the milk protein casein start to clump together and form yogurt. So what have we learned today? Today we gained the knowledge about anaerobic respiration by taking the example of yogurt formation from milk. So we have come to know that when organisms such as bacteria carry out respiration in the absence of oxygen, then such a respiration is called as anaerobic respiration. And fermentation occurs due to anaerobic respiration, and lactic acid fermentation is a type of anaerobic respiration carried out by bacteria, which helps us in yogurt formation. In your textbook, chapter 10, you might have seen it is written that the yeast forms alcohol in the absence of oxygen by anaerobic respiration. So you might be wondering whether yeast is the only organism that carries out anaerobic respiration. So answer is no. Bacteria and fungi, they carry out anaerobic respiration. Yeast is a type of fungus. And here we have discussed about bacteria, Lactobacillus bacteria, which carries out the formation of yogurt in an anaerobic condition created by another bacteria, Streptococcus thermophilus. So anaerobic respiration is useful for us, and we carry out fermentation of milk to produce yogurt, which is good for our health. So with this knowledge, now you have to search for few answers. This homework will test your understanding of today's lesson. So what is your homework? You have to go to chapter 18 of NCERT Science textbook, class 7. The chapter is wastewater storage, and you have to find out at what stage anaerobic respiration is used to treat wastewater. And you have to write the answer in your science copy. Then second question for your homework is, do we use yeast for anaerobic respiration? When and where? You may wonder where to search for the answer. Then you have to read chapter 10 from your NCERT Science textbook, class 7. So thank you for your patient hearing. Have a nice day.